Beyond Engineering, Design, Intelligence, and Instructions. When seen from the anthropomorphic perspective of engineering, design, intelligence, or instructions, the multicellular reproduction of complex structures and functions during animal development seems unimaginably challenging. In the absence of conceptual frameworks that are now widely accepted, the robustness of development remained a black box to experimental embryologists for about a century after Darwin published his On the Origin of Species. Peter Lawrence and Michael Levine provide a lucid explanation. From the mid-1800s, and for about 100 years, mainstream embryologists ignored genetics and tried to understand the mechanisms of animal development without it. The attempt was a brave one, but it became increasingly foolish. In the early 20th century, the gifted embryologist Thomas Hunt Morgan realized the importance of genes and took what he thought would be a temporary diversion into genetics. But it was a long detour. Only at the end of his life was he able to return to his beloved embryos. But most other embryologists continued to work, as if genes were irrelevant. Many of their results were so counterintuitive and conflicting that their hypotheses became abstract and ornate. The philosophical and the whimsical found this attractive. By contrast, it was the mathematical and the rigorous who joined the new science of genetics. Naturally enough, the two types of scientists failed to understand each other, and embryology drifted off into metaphysical swamps, while genetics explored the dry savannas of statistics. Escape from both the metaphysical swamps such as vitalism and entelechy and the dry savannas such as population genetics became possible when several brilliant scientists, including Ed Lewis, Christian Nusslein-Volhard, and Eric Wieschaus, applied Mendelian and molecular genetics to a systematic analysis of the development of fruit fly embryos. By relying on state-of-the-art methods such as in situ hybridization and other methods for visualizing the patterns of expression of RNA and proteins, these pioneers provided deep insights into the molecular, cellular, and developmental mechanisms of embryonic development. These mechanisms are determined by complex interactions between protein coding and regulatory DNA sequences that are now well understood. This has revolutionized our understanding of both evolution and development. More about this will be discussed in Chapter 15. In this context, the vitalism and entelechy of embryologists such as Driesch can be seen as well-meaning but misguided attempts to explain the astounding robustness of embryonic cells to form a complex and coherent individual. The tendency to seek supernatural explanations for phenomena that we do not understand is deeply embedded in the human DNA. But we also have the capacity to find scientific explanations for these phenomena, and once the facts are available, such supernatural explanations are often supplanted by scientific ones.